to set up this great conference. Yes, it's kind of unusual to see them in my office, but still, let's try. Okay, what I would like to discuss shortly with you today is basically two things in a small part. So the first part will be about the mitochondrial and aging as ancient partnership, and I have to say it's kind of philosophical thing, but I will show us certainly some data, but I would like to bring some new ideas how mitochondria might be engaged in aging and why is that. The second part is on the mitochondrial calcium uptake machinery, particularly in MAM regions. And I would like to show some few slides regarding the contribution of UCP2 in that. That will be the smallest part. And the third part is quite an interesting practical example of the ER mitochondrial crosstalk, particularly that is involved in pancreatic beta cell function. So let's start right away from mitochondrial aging, and let's start by a very simple question, which might be sound stupid, but I think it's not. So the question is, would you ever touch the sun? And I hope nobody would say yes. So that's basically why I address this question is because when you go in the literature, in particular, look in the literature of written by Gottfried Schatz, who is actually the name our research center is called Gottfried Schatz Research Center. He was calculating that hold somebody's hand and feel its warmth. Gram per gram, it converts 10,000 times more energy than 1 gram of sun. And I actually tried to recalculate that to the mitochondria and come out that 1 gram of mitochondria converts about 30 million times more energy per second than 1 gram of sun. What I want to say in that is now, of course, it indicates that these organelles are very, have a high energy density. And this is, we should not forget that maybe the mitochondria is not always the good guy. Maybe they can turn very bad. And the other thing I would like to share with you is how Austrians dies, but basically you can exchange that with every Western country, which is indicating that, so you see here on the left side, I think the men, right side, the women, and you see there's a certain, that first of all, most people die on cardiovascular disease. Sorry, it's in German, but it's in Austrian statistics, then in neural disease and cancer. But what puzzles me here in these figures is that we have an onset people die. And particular, so between 40 and 50, people start obviously dying. And this is becoming 57 this year. So for me, it's a big point here, you know. So I was thinking, is why suddenly people start dying and what is this aging associated death all about? So I was thinking, what is changing in this particular year between 30 and 50, let's say, and what came, unfortunately, to my mind that I became fat. So we call it big, but anyway, so people gain weight, particularly in this particular time of life. So, and this is now I would like to share with you a picture painted by a painter called Mr. Knight, and he painted that, and this is actually from the New York Gallery of Natural Science. And I think this picture, is very interesting because first of all, you saw the young fellows here. So you see the young lady. Maybe do you see my mouse? I hope you see the mouse, which indicates the young lady with the toddler and the young man with the weapon, indicating the young lady important, responsible for raising the kids and giving birth. And the second one is the young hunter, which is supposed to be a very athletic person. That's normal, everybody would expect that. But what I think is very interesting is the old fellow. And the old fellow, as you indicated by the gray hair, holds a firestone in his hand. He's obviously taking care on a fire, but not hunting anymore. But what I think is interesting, it still is athletic. So I was wondering whether there is an ancient adaptation that because of such a group of people might feed the young ladies and the young men very good food, and the old guys will not get enough food or maybe bad food. But this guy still remained slim. So in other words, maybe this person gained efficiency and still kept his figure, while we have still enough food, and that's the reason we become fat. So in other words, is there an ancient some metabolism switch that makes us more efficient? And having said that, I was thinking what makes animals, what makes cells more efficient? It's certainly the mitochondria. So when you compare the non-mitochondrial pathway of glycolysis, that roughly gives you 2 ATP, while the glucose metabolism that is coupled to mitochondria gives you around 30 to 40 ATP. So you have a gain on efficiency, and this is linked to ATP. And I was wondering, and still wondering, whether aging means also gaining efficiency by introducing more and not less mitochondrial activity in aging. So this is just more question than a fact. So in other words, you have a fast metabolism that maybe is due to young people that are delivering fast muscle contractions on. And maybe with age, we change our metabolism towards efficiency, meaning we have more coupled towards mitochondria. And surprisingly, it is the fact in several cell types, and actually many, many cell types we have tested. And I just give you here an example of human endothelial cells, that once when they're getting age, sorry for that, when they're getting age, you see the basal respiration is actually increased. So the lower curve is a freshly isolated endothelial cell, and after 5 passages on a model of aging, you see that the baseline is increasing of baseline respiration of these cells. And this we have found in many, many cell types, and indicating that aging is obviously really associated with an increased basal mitochondrial activity. Okay, having said that, how is that increase managed? And we have to go back to a little bit biochemistry, and we all know that that mitochondria depend on the Krebs cycle, and the Krebs cycle has 4 important dehydrogenases. And these dehydrogenases are dependent on mitochondrial calcium. So in other words, and you will see that in my later part as well, mitochondrial calcium not only fuels a lot of mechanisms in the cytosol, but it also fuels and stimulates the Krebs cycle. In particular, the first dehydrogenase, the isocitrate dehydrogenase, is an enzyme that under normal condition is only about 30 to 40% active, and essentially needs an elevation of mitochondrial calcium to get activated. So that, I think, is an interesting phenomenon, and I think the question is how the mitochondria gets the calcium. I think everybody in this group knows it very well. That is the MAM area, and that is the communication between the endoplasmatic reticulum and the mitochondria, and there's a lot of calcium shuttling happening. And we were testing this MAM area, or visualizing this MAM area by structural illumination microscopy. This is a kind of super resolution microscopy, and we calculated the interaction sites of the mitochondria with the endoplasmatic reticulum in aged and young cells. And you see, as on the left side here, that the communication between the organelles has been increased by about 30% during aging. And this increase is associated with an increased expression of MAM proteins like the RAB32, the RCAP1, or the IPC receptor type 1. So this somehow makes, for our point of view, a little bit sense. That means that in aged cells, the communication between the mitochondria and the ER is increased, and as a consequence, mitochondria are much more engaged in basal activity. I'm just talking about basal mitochondrial activity in these cells. And this would explain now this increase that we are measuring. Okay, so this was somehow this part, and I would like to sum up a little bit on this new aspect, I think, that indicate, and we found very interesting and very similar data in cancer as well. So we can say that during aging and cancer, the physical tethering between the ER and mitochondria is increased. However, it's always happening in aging. Some cancer don't do it. So we found about 40% of cancer cells have this increased tethering. More than 50% don't have. Then we can say an increased tethering yields elevated basal calcium that shuttles from the ER towards the mitochondria. And this increase of mitochondrial resting calcium stimulates particular the hydrogenases and stimulates citric acid cycle. And finally, this may, and this is important for cancer, just an estimation, that this may uncouple glycolysis. You remember the Warburg effect, uncouple glycolysis from the oxphos and introduce an alternative resource that's glutamine that goes here into essentially needs the hydrogenases to finally produce energy. So this is the first part. I would like, this is a very short informal thing, not some data included, but I would like to show you some of my thoughts how maybe mitochondria is engaged in aging, particularly by elevated tethering towards the ER. As the next second part, I would like to shortly review our work of UCP2 on mitochondria, because we have published some, actually 13 years ago, that the uncoupling proteins 2 and 3 are fundamental for mitochondrial calcium uptake. And you remember, I think that at these times, we didn't know that there's MCU or MICO1 or whatsoever. We just found in experiments that when we knocked down UCP2 or UCP3, we had a decreased the mitochondrial calcium uptake. And what was interesting in this story, and we didn't know a very long time what is happening, is that particular in this, if this is UCP2, the intermembrane loop 2 was the most important one, because if we exchange this one with the intermembrane loop 2 of UCP1, this protein was not active anymore. And importantly, this intermembrane loop 2 is that part where UCP2 and 3 differs most from all the other UCPs. So this makes UCP2 and 3 very unique. And we were puzzling because there were a lot of data, people couldn't reproduce this data, and we were puzzling for 13 years what is happening. And thankfully, we could finally solve this issue, and the issue is that obviously the engagement of UCP2 in mitochondrial calcium needs an effect that now we could solve, that is the methylation, the PRMT1. PRMT1 stands for protein arginine methyltransferase 1. And if MICO1 gets methylated by PRMT1, suddenly it is UCP2 and 3 is engaged. How this works? So here is a cancer type cell line. This is endothelial cells. I'm sure you know these EAHY926 cells. And what you see here is when we knock down UCP2, this is the red bar, you see this decrease in mitochondrial calcium uptake. That's always occurs. However, if we knocked out PRMT1 in the cell type, suddenly the mitochondrial calcium uptake was established and was insensitive to a knockdown of UCP2. Okay, so that was already an indication that maybe PRMT1 is doing something. And then we did the experiments vice versa. We took a very fresh isolated HUVEC endothelial cell and tested whether UCP2 has an effect on mitochondrial calcium. Again, this is the red bar, and as you see, it didn't have an effect. However, if we introduced now PRMT1 by overexpression, it didn't have an effect on the mitochondrial calcium uptake per se, but what happened if we knock down UCP2 in the cell that has overexpressed PRMT1, suddenly you got this decrease. So that was an indication that the engagement of the UCP2 is somehow involved, essentially needs PRMT1 activity. And we measured then also the calcium elevation in freshly isolated mitochondria. What we found out is that PMT1 or the PMT1 in a UCP-dependent mechanism seems to shift the calcium sensitivity of the uptake from 5 micromolar to about 14 micromolar. So in other words, without UCP, when PMT1 is present, without UCP2, the uptake has in KD that is about almost 3 times higher than when there's UCP present. So we were then luckily in 2016, or actually 2014, when we started this story, there were already a lot of proteins known from the mitochondrial calcium uniporter, thanks to the work of really fantastic guys. And so we were thinking, we test what is PMT1 doing, and we tested all of them that were known at that time particular. And we found out that MICO1 is getting methylated by PMT1. So as a next step, we were wondering, we can measure the calcium sensitivity of MICO1 by simply using FRET. You have a CIFB, a YFB of MICO1, and you regulate by calcium its deregulation or dearrangement. And we have done so with the MICO1, as you see here. And when without PMT1, there was no change in the sensitivity of MICO1. However, if you had PMT1 activity present and removed the UCP2, suddenly the calcium sensitivity of MICO1 was changing, and actually changed in a pretty much the way the calcium uptake changed. In this case was from 4 to 18 micromolar. So in other words, this work indicated that UCP2 is engaged in the moment when PMT1 activity methylates MICO1. And this is happening in particular in aging and in particular in cancer. Okay, so, and this is the last story, last part of the UCP reminder, so that we have recently shown that MICO1 controls crystal junction. That's a very new function of this protein, and we see here that this SIM microscopy data, and you see here in green you see the MICO1, and in what is this, magenta, you see the mitochondrial dye, and you see that MICO1 seems to be building a shelf around the mitochondrial matrix. And this is where we have tested that with Tom, and luckily we have the super resolution microscopy. We could finally find out that indeed MICO1 is in the inner mitochondrial membrane, but obviously does not enter the crystal. And exactly the opposite is happening with the MCU complex. Here you see the MICO1 in green having this beautiful shell that you see in the other slide as well. And the MCU is somehow in the matrix. Certainly it's not in the matrix, but it is in the crystal. So MCU mainly is in the crystal, but once when you get stimulated by an agonist, in this case it was histamine, MCU shifts towards the, somehow joins the MICO1 in the inner boundary membrane, so goes out of the crystal into the inner boundary membrane and stays there. And then after the signal goes down, you see in here that it goes back into the crystal. And this happens in simultaneously. So the lower one is a simultaneous recording of the localization we see up there, and the mitochondrial calcium. And you see that mitochondrial calcium slightly follows the relocalization of the MCU. And I apologize, there's no better time resolution possible with the MCU localization. There's a lot of shifting happening. So in other words, we could find that MICO1 controls is sitting in the outer side of the inner mitochondrion. This is the inner boundary membrane. And the MICO1, once activated, seems to float there and to communicate. And exactly this mechanism, and I show you only the graph here, was dependent on UCP2 once when PMT1 was active. when you see in the left side of the graph of the scheme, you see when no PRMT1 is there and UCP2 is also not there, nicely the MICO1 catches the red MCU embryo complex and allows entry. However, if PRMT1 is active and UCP2 is missing, then this MCU complex does not, this MICO1 MCU complex does not establish, and MCU and embryo remain in the crystal. Once when you add UCP2 in the system, then UCP2 helps to catch the MCU and the embryo complex and establish calcium entry. So that was somehow so far our results. What is the UCP doing? And all looks like that everything depends on the arginine methylation of the MICO1 so far. And another important aspect, I don't want to go too much in detail, but I think it's at the moment, it's one of our focus in research, is that MICO1 plays a very similar role than OPA1, meaning it's the opening of the crystal junction. But importantly, MICO1 is the calcium-dependent opening of the crystal junction. So while OPA1 is not regulated by calcium, MICO1 is calcium-regulated, and we know in our recent work that calcium can open crystal junction by an activation of MICO1. And also here, what a surprise, when PRMT1 is active, UCP2 is also playing a role. So this is just a short interplay regarding our UCP2, and I just want to go to the last part. It is a kind of practical indication how the ER mitochondrial crosstalk is involved in pancreatic beta cell insulin secretion. Shortly, please allow me to share the concept of insulin, regulation of insulin secretion. And we all know that there's glucose uptake, and this glucose undergoes glycolysis, and the acetyl-CoA goes then finally the citric cycle, forms citric acid and goes to the formation finally of the NADH and FADH2, and this is converts to the OXPHOS, making ATP, and ATP, of course, is transferred to the cytosol via ANT, inhibiting the KATP channels, resulting in depolarization, opening of the L-type calcium channel, and the calcium influx occurs, and this uses goes to vesicle fusion and insulin secretion. So basically, roughly, this is the regulation of the ATP, of the insulin secretion. However, what I think is interesting is that people have found that mitochondria is function involved, of course, because of its ATP formation. However, there's one problem. As you see here in this slide, and as we discussed earlier, the calcium citric cycle, the dehydrogenases of the cycle, this time it's on the other side around, are calcium dependent. So the question is, when these proteins are calcium dependent, how is calcium important later on? And we know particular, as I said before, the isocitrate dehydrogenase, meaning the first dehydrogenase needs essentially an increase of basal calcium. And the importance of mitochondrial calcium elevation has been published. However, it is always indicated not regarding the EDH, but indicated regarding the ATP and calcium here. So my question is there a process that ensures proper elevation of mitochondrial calcium? Because this one comes first before the calcium enters. So once when we need the calcium elevation here to finally increase cytosolic calcium, we must have a mechanism that ensures basal calcium elevation already before the calcium enters here. So somehow the concept, from my point of view, there was something missing, and we wanted to study that. Okay, so, and when we started with beta cells, and I guess many of you have this experience as well, I was really pretty much surprised that beta cells have a huge ER calcium leak. That is really impressive. And when you put them in zero calcium, they run out, the ER runs out of calcium, as you see here. And interestingly, when you compare that with other cell types, there's no or little ER calcium leak. So for us, that was very interesting. And I can tell you, I don't have time to go in detail, but I can tell you that this loading decision, so to keep the ER calcium high in the beta cells is achieved by the TRPC3 activity. So when you looked on the ER calcium, so this is now ER calcium measurements, you see again, when you remove extracellular calcium, the ER gets depleted in the beta cells. So these are two beta cell lines. However, nobody knows actually, from my point of view, it was for us not clear how much was this depletion. So we did experiments, and as follows: we allowed the beta cells to keep the ER calcium high, meaning there was extracellular calcium, and then we depleted the ER completely by BHQ and carbachol. And this we compared then with a cell that was where we removed the extracellular calcium and kept it 18 minutes in zero calcium. And as you see here, in about 18 minutes, we reached about that point, meaning the ER calcium leak that we see here on the other side is a very, very strong ER calcium leak. It's not a minimal, it's really a very considerable calcium leak. For us, that was already important to think about, does this, as this is so specific for beta cells in our hand, we were wondering whether this is an important feature for these cells and whether it plays a role. Okay, so we were checking literature about the possible ER calcium leak channels, and for us, presenilin 1 particular came as we tested as the most prominent one. And as you can see here, when we knock down, these are the red lines, when we knocked down presenilin 1 in the beta cells, indeed the calcium leak was gone. When we overexpressed presenilin 1, we had even a little bit more calcium leak. But most importantly, I think knockdown almost completely recovered the calcium leak. So, of course, it was great. We were very excited. However, all cells, so even the cells that didn't have the calcium leak, had presenilin 1. So that was a bit frustrating for us, because now it was not a simple answer to us that presenilin 1 is the only thing. And then my thesis student, Ms. Kleck, realized that GSK3 beta is known to stimulate the calcium lead of presenilin 1 by phosphorylation. So we tested now phosphorylation activity, GSK3 beta phosphorylation activity, and here we came to at least a significant higher GSK3 beta activity in the beta cells than with our compared non-beta cells. Okay, so when that is true, that GSK3 beta mediated presenilin 1 establishes the ER calcium leak, that's a very simple thing. What you can do, you prevent GSK3 beta by inhibitors, and there's luckily one inhibitor available. You can't do knockdown because the cells is going to die. So you, when you do acute inhibition, you see that here that the calcium leak is completely abolished in both cell types you use. So this, and more importantly, also in isolated pancreatic islets. So obviously, even in isolated pancreatic islets, preventing GSK3 beta removed the ER calcium leak. So we played the other way around. So we expressed an PS1 presenilin 1 mutant that prevents phosphorylation of GSK3 beta, and as you see here, that the calcium leak of the cells suddenly was diminished, also indicating then that in fact GSK3 beta mediated phosphorylation of presenilin 1 really introduces the calcium leak. And this is our next control. We expressed in our cells that didn't have the calcium leak, these are HeLa cells and EA cells. We expressed a presenilin 1 mutant that mimics a phosphorylation by GSK3. beta, and you see we introduced the calcium leak in the ER by introducing a mediated phosphorylated presenilin 1. For us, this indicates now that obviously GSK3 beta in the beta cells phosphorylates presenilin 1 and establishes a very, very considerable ER calcium leak in the cell type. So what are the consequences of this ER calcium leak? This is continuously and becomes obvious only when you remove extracellular calcium, but it's continuously there, but it's fueled by extracellular calcium. You see here, mitochondrial calcium is significantly elevated in this cell type. However, you know, this might happen to many cells. So the other thing we tested now, what happened when we knock down the MCU in the cells. And when we knock down MCU, cytosolic calcium increases in the beta cells, but didn't do so in other cell types. So this indicates, obviously, already indicates that their calcium leak is maybe directed towards the mitochondria and is taken up by an MCU-dependent phenomenon. So, and this MCU-dependent phenomenon was gone when you prevented phosphorylation of the presenilin 1, as you see here, or removed the presenilin 1. Then there was no calcium leak and consequently no effect by knockdown of MCU. Okay, as a final proof that under this condition of normal condition, that there is a small calcium flux from the ER towards the mitochondria. We measured mitochondrial calcium, and as you see in the upper image, that a removal of extracellular calcium rapidly decreased calcium concentration in the mitochondria as an indication that this flux is now reduced, while it didn't so in the HeLa cells. So far it looks like that everything worked fine, that there's really a calcium leak that is kind of directed towards the mitochondria in beta cells. So what is the physiological consequences? And I would like to show you now respiration of beta cells. This is pretty high, as you can imagine. But if you knock down presenilin 1, the basal respiration was reduced. And this is happening by knockdown of presenilin 1, as well as by inhibition of GSK3 beta. So again, this proves that this concept of calcium going from the ER towards the mitochondria in a basal condition to elevate basal activity of mitochondria and beta cells holds true also when we look to respiration. It also was shown that ATP, resting ATP levels in the beta cells was increased due to this activity, as you can imagine. And when we knock down the presenilin 1 or prevented GSK3 beta activity, so these are this inhibition that you see here, then you see also an ATP reduction. As in proof that this shuttling of calcium on a basal condition had a physiological increase of ATP and OXPHOS activity in the beta cells. So what was, of course, now in physiological meaning, there's one important thing. This in beta cells, this is glucose-induced cytosolic calcium spiking. And as you see here, when you add high elevated glucose, these beta cells start cycling, have this nice spiking. When you do so by preventing presenilin 1, the spiking was decreased, as well as by inhibition of GSK3 beta. So again, this indicates that there's a physiological consequences when the calcium transfer towards the mitochondria is not happening. Seem to be that the beta cells are kind of reluctant to glucose. And finally, we did the ultimate test was, of course, the glucose stimulated insulin secretion. And you see here that knockdown of presenilin 1 in an insulin-normal cell line inhibited this early phase of insulin production, this initial phase, while insulin content was not affected by presenilin 1, and the same effect we found out by inhibition by GSK3 beta in the pancreatic islets, in freshly isolated pancreatic islets. So summing up that, it looks like that beta cells have a very, we have another point of the regulation of insulin secretion in beta cells, and this is particular how the cytosolic cycle is has its capacity to really sense the increase in glucose, and this is achieved by a mechanism that calcium goes from the ER via the phosphorylated presenilin 1 and a basal condition to the mitochondria. Calcium elevation increases cytosolic activity, and under this condition, then obviously glucose can be better sensed in this insulin. And we were thinking, is that affected also? Can we see something in human? And I think it's quite interesting that when you go in diabetic papers, you see that there's a so-called non-diabetic state where the first phase of insulin release in human has been measured, and there's a phase in people most likely will develop type 2 diabetes. There's no initial phase. And finally, I would like to end that it's the way that obviously Alzheimer is also linked to that as well, of course, presenilin 1, but also to diabetes. And I found a paper, they indicated even Alzheimer's disease as type 3 diabetes. And it might be interesting to that maybe presenilin 1, this shuttling, is not only involved in Alzheimer, but also in development of type 2 diabetes. Just shortly, the team. This is my team in our new buildings. And these are our collaborators, our organizers and companies that support us. And finally, I thank you very much. Thank you.
Okay, great. Thank you very much.